too.
Yes. Welcome, everyone. Welcome to our folks here at the UW and Harborview and Children's and everyone else who's watching from their own personal console. It is my great pleasure to introduce Dr. Ian Mackenzie, who visits us from UBC, just up across the border. And so Dr. Mackenzie is a professor of pathology at UBC in medicine, and he did his MD and residency in Ontario and a research fellowship in Australia in pathology. And over the years, Dr. Mackenzie has led his team and his colleagues to really opening up the landscape of our understanding of FTD and neurodegenerative disease. He has helped to characterize by pathology different phenotypes and subtypes of FTD, and that has really led the way for us to understand more about the different genetics, different mechanisms, and he has been a part of identifying TDP43 as an important member of the inclusion bodies, identified genes such as progranulin, and most recently C9orf72. So really, my 1-minute intro does no justice, but it's really exciting to have him here and give us some information. Thanks.
Thank you. Well, thank you for the invitation to speak with you this afternoon. Bring this on. And really what I'm going to do today is just provide you with a fairly broad discussion of the molecular basis of FTD, emphasizing some recent advances that have been made over the last 5 or 6 years. And what I'm going to do is present this largely in sort of chronological order, because I think it makes a nice story, for one thing. It also illustrates the rapidity with which this field has evolved over the last few years. And also it's the only way that I can think of to show how the discoveries related to FTD have been so intertwined with those related to ALS, to the point that these two disorders are now often considered to be part of a spectrum of disease. And it's amazing how identification of a gene in one disease has then led to an advance in the other disease, and vice versa. It's almost sort of flip-flop back and forth. And it's hard to do if you simply try to focus this on sort of the current view of things. So this is going to be a little bit of a temporal story. So first of all, frontotemporal dementia, as you know, it's a clinical syndrome defined by progressive changes of behavior, personality, and language with relative early preservation of memory, and we're actually starting to get away from that a little bit. And it's broadly subdivided into those that present primarily with behavioral abnormalities and those with primary progressive aphasia based on the presenting phenotype. Having said that, these phenotypes do tend to converge as the patients advance, so often they will end up with both language and behavioral abnormalities. Although FTD is not nearly as common as Alzheimer's disease, it is nonetheless a very important dementia subtype. Probably accounts for between 10 and 15% of all dementia. And I think its importance, its societal importance at least, is increased by the fact that it tends to affect slightly younger individuals. Mean onset is about 60 years of age, but some actually onset in their 30s and 40s, so at an age when they're raising their families, still very active in their jobs, for instance. So it has an even greater societal impact. It also has a bigger impact in a way because it has a stronger genetic basis. So a lot of these cases have a familial tendency, and up to 25% are autosomal dominant. And what that means is not only is there obviously a strong genetic component, but that the diagnosis of FTD then has major implications for other family members. And then finally, FTD is often associated with movement disorder, either an extrapyramidal or pyramidal movement disorder. And so research in FTD is now beginning to overlap more and more with research in ALS and research in Parkinsonism. Now, the neuropathology that underlies the clinical syndrome of FTD is heterogeneous. A consistent feature is the relatively selective degeneration of the frontal and temporal lobes, which of course corresponds with the behavioral and language abnormalities. And the term frontotemporal lobar degeneration is commonly used as a term for the pathologies that may underlie clinical FTD, and that's how I'm going to use the term for this talk. So FTD for the clinical phenotype, FTLD for the pathologies that are related to FTD. In addition to non-specific changes of neuronal loss, reactive gliosis in the affected areas, most cases of FTD at pathology are found to have some abnormal accumulation of protein in the form of inclusion bodies in the tissue. And until quite recently, the only major group of these FTLDs that we knew anything about the molecular basis were those characterized by the abnormal accumulation of the microtubule-associated protein tau. And these include cases with the pathology of classical Pick's disease, corticobasal degeneration, progressive supranuclear palsy, and all of the families with autosomal dominant FTD caused by mutations in the MAP-tau gene. I'm not going to talk in any detail about these tauopathy cases because they're probably the ones that you're most familiar with. They've been known about for some time. They've been fairly well characterized, and you can read about them in most standard textbooks. Instead, I'm going to focus more on the other subtypes of FTD and the more sort of recent discoveries that have affected those fields. So in fact, most cases of both sporadic and familial FTD are not associated with tau pathology. And until fairly recently, it was felt that these other cases, these non-tau cases, did not have any identifiable protein abnormality, and they were sometimes given this horrible term of dementia lacking distinctive histopathology. However, an important advance in our understanding of these non-tau-based FTDs came initially from a discovery in patients with ALS. So as you hopefully know, patients who present with ALS, about 10% of them go on to develop full-blown dementia, which often has the clinical features of FTD. And in fact, up to 50% of ALS patients have more subtle frontal lobe dysfunction that can be demonstrated with neuroimaging or neuropsychological testing. And in the early 1990s, a novel type of pathology was first discovered in these patients with ALS and dementia in the form of these neuronal cytoplasmic inclusions and dystrophic neurites in the upper layers of the extramotor neocortex and the hippocampus that at the time could only be demonstrated with immunohistochemistry for the non-specific marker of protein accumulation ubiquitin. Shortly after this was discovered in the ALS dementia patients, various groups started reporting similar ubiquitin pathology in subsets of patients with clinically pure FTD with no motor abnormality. And in that context, the terminology FTLDU for ubiquitin, or some people used it as unknown, became the phrase for that pattern of pathology. And although it took about a decade, with growing appreciation amongst neuropathologists and with improved immunohistochemical techniques, it eventually became apparent that most of these non-tau-based cases did in fact have this FTLDU pathology, and that this was in fact the most common pathology underlying clinical FTD. Moreover, as we began to accumulate large numbers of these cases, it became apparent that there were different patterns that we could recognize based on the morphology of the inclusions and their anatomic distribution. And what made us think that there was something important to this was there seemed to be fairly specific clinical correlations between these patterns and the associated phenotypes. So in this classification that David Mann and I came up with, the cases we refer to as type 1 pathology, where there's lots of neurites and cytoplasmic inclusions, largely confined to lamina 2 of the neocortex, these patients all tend to present with either behavioral variant FTD or progressive non-fluent aphasia. In contrast, our type 2 cases, characterized by these very long neurites and virtually no cytoplasmic inclusions, they almost always have semantic dementia clinically. And all of our patients who had a combination of FTD and ALS turned out to have our type 3 pathology, where there was a predominance of cytoplasmic inclusions and very little neurites. Moreover, it turned out that each of the newly discovered FTD-causing genes that came along in the next couple of years were each consistently associated with one of these specific patterns of pathology. So in 2006, collaborative research between our group at UBC and Mike Hutton's lab at the Mayo Clinic in Jacksonville identified progranulin as the second FTD-causing gene on chromosome 17. Progranulin is a multifunctional secreted growth factor. It's produced in a variety of tissues throughout the body, but in the brain primarily by neurons. The first mutation that was discovered was in our family UBC17. It's an insertion in exon 1 causing a frame shift. There have now been over 70 different pathogenic progranulin mutations identified in over 200 families worldwide. making this a more common cause of familial FTD than even the MAPTau mutations. As you can see, the mutations are of various sorts and occur throughout the gene, but they all have a common effect. They all generate a null allele. Basically, they silence the mutant allele. And so disease in these patients is not caused by the production of a mutant protein, but instead by the silencing of one allele and the knockdown in the amount of normal protein being produced, so a so-called haploinsufficiency mechanism. The disease is autosomal dominant, it is highly penetrant, and the phenotype is usually either behavioral variant FTD or progressive non-fluent aphasia. Very often these patients also develop some degree of Parkinsonism, although it's usually mild. It usually onsets after the FTD is already established, but it can be fairly significant, and it can even look like a full-blown corticobasal syndrome or rarely a PSP-like syndrome. But interestingly, these patients don't develop ALS. And that will become a more interesting finding as we move on and discuss the eventual molecular basis of this. The pathology in these cases is fairly consistent. They are all characterized by what we had originally described as our FTLU type 1 pathology, where you've got lots of neurites, lots of inclusions in the superficial neocortex. But they have one additional feature, which is the presence of moderate numbers of these interesting lens-shaped or lentiform neuronal intranuclear inclusions. And these are a good clue when you look pathologically at a case that there is an underlying either mutation or genetic variant within the progranulin gene. Now, around the same time, so we're now at 2006, in 2006, families started to be reported in which the affected individuals developed either ALS or FTD or a combination of both. And in the pathology that was described in some of these families, it was consistently what we had called our FTLU type 3, where there was a predominance of these cytoplasmic inclusions. And the ones that had ALS also had the typical ALS type inclusions in the lower motor neurons. There's also this large Danish pedigree with FTD linked to chromosome 3 caused by mutations in the CHIMP2B gene. Ida Holm and I have looked at all of the pathology available from this family, and it consistently shows these ubiquitin-positive inclusions that are largely restricted to the hippocampus. So this fits with our FTLU type 3B, B simply meaning that it's restricted to that one anatomical area. And then finally, there's this rare familial syndrome of inclusion body myopathy with Paget's disease of bone and frontotemporal dementia caused by mutations in the velocin-containing protein gene. Mark Foreman and I had an opportunity to review a series of these cases provided by Virginia Camonis, and we identified a new subtype of FTLDU characterized by all of these very delicate little neuritic processes and many of these intranuclear inclusions with almost no cytoplasmic inclusions. So again, these different patterns are based on the cellular localization of where the protein's accumulating and what lamina of the cortex are involved. But what wasn't known at this point, so this is sort of mid-2006, was it wasn't known if the ubiquitinated protein in all of these different clinical and genetic disorders was the same, or you'd probably more likely assume that they were different, or there were at least some different proteins. However, this was largely resolved late in 2006 when work in Virginia Lee's lab at University of Pennsylvania, led by Manuela Neumann, identified the transactive response DNA binding protein molecular weight 43, or TDP43, as being the ubiquitinated pathological protein in most of these major subtypes of FTLDU. TDP43 is a DNA RNA binding protein. It's involved in many different aspects of RNA metabolism. It's normally localized to the nucleus, but it does shuttle back and forth between the nucleus and the cytoplasm. In addition to showing that the ubiquitin-positive inclusions in all of the major FTLDU subtypes also co-label with commercial antibodies against TDP43, Virginia and Manuela also showed that the inclusions are composed of an abnormal C-terminal fragment of TDP43 that is hyperphosphorylated and ubiquitinated. And at a functional level, an interesting finding is that all of the cells that contain these TDP43 positive inclusions lose their normal nuclear staining. So the normal localization to the nucleus is somehow lost. Again, possibly implying some sort of loss of function mechanism in disease. Now, because of this perception that ALS and FTD were somehow related conditions, In this initial study, they also looked at a small number of ALS cases and found that the ubiquitin-positive inclusions that are characteristic in lower motor neurons of ALS are also composed of TDP43. And so this finding of a common molecular defect in most cases of ALS and in the most common subtype of FTLD was the strongest evidence to this point, confirming our suspicion that FTD and ALS were somehow related disorders that shared at least an overlapping molecular basis. And our initial, somewhat misguided or optimistic view was that TDP43 pathology was an absolutely specific and sensitive finding for this spectrum of disease. But that quickly started to fall apart. And in fact, just the following year, we published this much larger study of over 100 ALS cases in which we found that the cases associated with SOD1 mutations do not have significant TDP pathology. So if you look at the bottom here, the top 3 rows are sporadic ALS cases without SOD1 mutations. The bottom 3 rows are 3 different cases with different SOD1 mutations. What you can see is that they do in fact have ubiquitin-positive neuronal inclusions stained with green immunofluorescence, but these do not co-localize TDP, and in fact the TDP retains its normal nuclear localization. Moreover, these cases with SOD mutations lack the lower molecular weight bands, the disease-specific bands on immunoblotting. And in fact, we now know that these ubiquitin-positive inclusions in the SOD1 cases are, as you might expect, composed of misfolded SOD1, as you can see here. So this finding of an absolute biochemical distinction between the ALS caused by SOD1 mutations and the sporadic cases, I think, was important in that it suggested that the mechanism of neurodegeneration caused by SOD1 mutations was perhaps different than it was in sporadic ALS, implying that SOD1-based therapies and models might not be appropriate for sporadic ALS. And this was not well received by the ALS research community, who had spent the previous 10 years devoting 99% of the research to SOD1-based mice and SOD1-based therapies. And in fact, there's still a lot of resistance to this. However, those were the findings. Now, the following year, 2008, we also recognized that about 10 to 20% of the cases that we had originally classified as FTLDU did not have TDP pathology. Most of them did, but a small proportion didn't. And I coined the unfortunate term of atypical FTLDU for these cases because they run atypical both clinically and pathologically. So clinically, they have a very unusual and very consistent pathological phenotype. They are all sporadic. They have very early age of onset. I've got one patient who was 26 when he onset. Most of them are in their 30s and 40s. They present with severe, rapidly progressive psychobehavioral abnormalities, often antisocial, even criminal behavior. They shoplift, they fight with their boss and get fired. They're involved in hit and run accidents, and they're rapidly progressive, so that all of the patients in our initial study were permanently institutionalized within 2 years of their initial disease onset. They were almost all initially diagnosed as being schizophrenic or some variant of major psychosis until the rapid progression became evident. They're also atypical clinically in that they don't develop the language disorder or the movement disorder that is fairly common in other FTD subtypes. And at a pathological level, they're atypical because their ubiquitin-positive inclusions are not TDP, and also they have this unusual type of intranuclear inclusion, which I'd never seen before, these very elongated, sometimes twisted, sort of three-dimensional, wiry intranuclear inclusions. I use the term vermiform for them. Again, I hadn't seen these previously, and these are an absolutely consistent finding in these cases. So what this implied, so the identification of this sort of new subset of FTLD cases told us that there was at least one FTD-related protein yet to be identified. And a clue to that came just a few months later, in early 2009, when these 2 studies were published, identifying mutations in FUS as a cause of familial ALS. FUS stands for fused in sarcoma, recognizing the fact that it was originally discovered because chromosomal translocation of the 5' region of FUS generates various fusion oncogenes that cause human cancers like myxoid liposarcoma. So that's where it was originally identified. FUS is also a DNA-RNA binding protein. It has high functional homology to TDP43, which is in fact why Bob Brown and Chris Shaw's group originally targeted it in their research. Like TDP43, it is primarily localized to the nucleus, but shuttles back and forth. The mutations that cause ALS in FUS tend to affect the C-terminal region of the protein. They're almost all missense mutations or deletions that affect the C-terminal region, which includes the nuclear localization signal, which is critical for the transportin-mediated nuclear import of the protein. And a number of in vitro studies have shown that the degree to which a particular mutation interferes with FUS binding to transportin directly correlates with the severity of human disease related to that mutation. So there's a good correlation that not getting FUS into the nucleus is related to how bad the neurodegeneration is. I'll just show you this. This is a family we published shortly after the mutation had been identified. It's an unusual family. It's a mother and a daughter. Their onset was unusual in that it was proximal weakness, originally diagnosed as a myopathy, and only later in the course of their disease did they develop more distal weakness and upper motor neuron features more consistent with ALS. But what was unusual is that they were living together and they developed the disease at the same time. Not at the same age, but within weeks of one another, suggesting a possible environmental trigger on a susceptible genetic background. Both were found to carry the R521C mutation, which is the most common ALS-causing FUS mutation. And pathology was available on the proband, who was the mother. And what we found was an absence of any TDP pathology, but lots of different kinds of FUS immunoreactive neuronal cytoplasmic inclusions, and also some FUS-positive glial inclusions as well. And this is fairly typical of the cases that have been reported with FUS mutations. So again, because of this perceived overlap between ALS and FTD, this led Manuela Neumann and I to wonder, could some of the remaining tau TDP negative forms of FTLD actually be related to FUS in some way? And in a series of experiments that we published later in 2009, we found that this was in fact the case, that virtually all of the remaining FTLDs that had been this sort of heterogeneous grab bag of things all turned out to have abundant FUS pathology. And these include our cases of atypical FTLDU, where you can see that all of the inclusions that were stained with ubiquitin also stain for FUS. And in addition, FUS stains more pathology, including glial inclusions that we hadn't seen with ubiquitin. There's a second condition, fairly uncommon, probably heterogeneous condition referred to as basophilic inclusion body disease, characterized by these round neuronal cytoplasmic inclusions which are basophilic or blue on a standard H&E stain. We showed that those basophilic inclusions are in fact contain FUS. And then finally, neuronal intermediate filament inclusion disease, which is another fairly uncommon neurologic disorder. It typically manifests as early onset sporadic FTD, almost always with a movement disorder associated with it, either Parkinsonism or ALS, and originally recognized by the presence of neuronal inclusions that contain class 4 intermediate filaments, which are neurofilament and alpha-internexin. What we found was that in fact these cases have much more abundant FUS pathology. So there are some inclusions shown in these top three panels that are only composed of FUS, no intermediate filaments. But what's more interesting is that in the bottom three rows, what you see is every cell that contains an intermediate filament positive inclusion also contains a FUS positive inclusion. Interestingly, they don't they don't overlap within that cell. They're sort of discrete regions within the same cell. So this led us to believe that in this condition, that the FUS pathology was more central to the disease. It started first, and that in certain subsets of neurons that were metabolically susceptible, they secondarily developed the intermediate filament inclusion sort of on top of the FUS inclusion. Not sure if that's right or not, but that's our working hypothesis. So with that discovery, it suddenly became possible to assign virtually every case of FTLD to one of three major molecular categories: tau, TDP, or FUS. Now, it gets a little bit more complicated because FUS is in fact a member of a family of proteins referred to as the FET family of proteins, FET simply standing for FUS, Ewing sarcoma, and TAF15. These proteins are all DNA-RNA binding proteins, and they were all originally discovered because of their ability to cause to form fusion oncogenes related to certain kinds of human cancer. And what we found in a subsequent study was that all of the different kinds of inclusions in all of these different FTLD FUS subtypes also label for the other FET proteins, for TAF15 and Ewing sarcoma. So in this immunohistochemical panel here, you see AFTLDU cases in the upper right, a NIFID case in the lower left, and BIBD in the lower right, all showing the inclusion staining for TAF15. And here, using double label immunofluorescence, we're seeing each of the different conditions with inclusions labeling for FUS in red and co-localizing Ewing sarcoma protein in green. So these are in fact not just FUS, they're all of the FET proteins. But what was really interesting in this study was we also looked at cases of ALS with FUS mutations, and in these cases, the inclusions only contain FUS. They don't contain any TAF15 or any Ewing sarcoma protein. Moreover, the FTLD cases, the inclusions also co-localize transportin, the carrier protein that shuttles the FET proteins in and out of the nucleus. However, when we looked at a number of other proteins that are normally carried by transportin, they don't co-localize with the other FUS or FET inclusions, and they show normal cellular localization. They manage to get in and out of the nucleus okay. And of course, when we looked at the ALS cases, the inclusions that are only FUS also don't contain the transportin. They're exclusively FUS. So what this led us to was this appreciation that the process of inclusion body formation in ALS with FUS mutations and sporadic FTLD FUS was distinct. There were two different processes going on here. That in the ALS cases, the mutations specifically interfere with the interaction of the FUS protein with binding to transportin, such that FUS then gets excluded into the cytoplasm where it aggregates and forms inclusions. The other FET proteins are fine. They go in and out of the nucleus. In FTLD FUS, there's some more general defect that interferes with all of the different FET proteins binding normally to transportin. And because the other transportin cargoes seem to have no problem getting in and out of the nucleus, it seems unlikely that this is a primary defect in transportin, a mutation in transportin, or just decreased expression of transportin, and more likely that it relates to some common abnormal post-translational modification of the FET proteins that's interfering with this interaction. And fairly recently, work done by Christian Haas provided evidence that the problem in these FTLD cases was one of methylation, was one of arginine methylation. And what they showed was that in all of the FET proteins, I'm just showing FUS here, but in all of the FET proteins, immediately adjacent to the nuclear localization signal is an arginine glycine-rich region. And they showed that it was also capable of binding to transportin, but that this interaction is normally inhibited by methylation of the arginine residues. And so they generated antibodies against methylated epitopes of FUS and the other FET proteins. They showed that normal FUS is methylated, and the normal FUS that's in the nucleus is methylated, as is the FUS that accumulates in the cases of ALS where the problem is the mutation in FUS. It's also normally methylated. But that the FUS that accumulates in the FTLD cases is not methylated, or at least is not normally methylated. It's hypomethylated. We don't know why at this point, but that seemed to be a consistent feature. And so the working hypothesis now is that arginine methylation of this arginine glycine-rich region regulates the binding avidity of the FET proteins to transportin, which normally occurs primarily through the nuclear localization signal. For some reason, in FTLD FUS, hypomethylation allows this more avid binding through both the NLS and the arginine glycine-rich region, forming complexes that cannot dissociate. And so you get the FET proteins bound to transportin that can't be released once they get into the nucleus. And they end up aggregating in an abnormal fashion. And so you get these inclusion bodies that contain everything. They include FETs, all FETs, and transportin. In comparison, in the ALS cases, the mutant FUS, which is normally methylated, can't bind normally to transportin because of the mutation. And so you get normally methylated mutant FUS accumulating in the cytoplasm. And as sort of further proof of this hypothesis, Dorothy showed that by experimentally blocking the methylation of mutant FUS, you could actually reestablish its ability to bind to transportin, now through this sort of aberrant site, through the arginine glycine-rich region, and allow at least it to partially relocate back into the nucleus. So I think a nice sort of biological proof of this hypothesis. Why the FET proteins are hypomethylated in the FTLD cases, we still don't know. The only thing we've been able to sort out is that Rosa Rademacher has looked at the genes encoding the responsible methyltransferases and has not found any genetic variability to explain it. So there doesn't seem to be a mutation in the mechanism responsible for that methylation. So we still don't know what that underlying mechanism is. So because of all of that, we fine-tuned our molecular classification a bit. And in the case of the FTLDs, we now refer to it as FTLD FET, because to be honest, we don't know whether one or the other of these FET proteins is more important or not than FUS. Whereas ALS, it's still purely FUS. Okay, I'm going to finish my talk with by focusing on what I think is by far the most exciting recent discovery in this field, which is the identification of the gene on chromosome 9p responsible for both FTD and ALS. And as I mentioned earlier, starting in 2006, a number of families were reported in which affected members could develop either FTD or ALS or both, and they seem to show genetic linkage to a common region on chromosome 9p. And the region where this linkage overlapped amongst the published families defined a common candidate linkage region that contained only 10 known or putative genes, of which only 5 are protein encoding. So a very gene poor area, just what you want to find as a geneticist. Interestingly and importantly, the same genetic region had been identified in several large independent GWAS studies of both FTD and ALS, implicating a genetic abnormality at the same chromosomal locus in the cause of both sporadic ALS and sporadic FTD. So that was interesting supportive evidence for this region. Despite this being a very gene-poor region, several years of exhaustive analysis, which in our collaborative group was done by Rosa Rademacher, but being done by many different groups around the world, failed to identify any mutations, altered splice patterns, deletions, duplications, altered gene expression. And so we knew that this was some weird kind of mutation. It wasn't something common and easy to identify. And the eventual discovery that the mutation was in fact abnormal expansion of a hexanucleotide repeat in a non-coding region of the chromosome 9 open reading frame 72 gene was made in 2011 in the lab of Rosa Rademacher. And this discovery largely was based on recognition of an apparent non-Mendelian pattern of inheritance in our family VSM20. So this is a family that we had originally identified in Vancouver. It then turned out that a branch of the family was also being followed in San Francisco and another branch at the Mayo Clinic. Luckily, we were already collaborating with all of these centers, and so this family became known as Vancouver, San Francisco, Mayo Family 20. And what was interesting is when you analyze these samples using standard fluorescent fragment length analysis, the first thing that is odd is that it looked as if all of the affected family members were homozygous for this polymorphic repeat region. And although that's certainly possible for somebody to be homozygous, it became increasingly unlikely as we looked at more and more specimens from this family. And in addition, it appeared that affected children were not inheriting an allele from their affected parent. So if you look at this limited branch of the pedigree over here, what you see is that the sample from the married-in unaffected husband appears to be is heterozygous, and I can't read that, but I think it's 5 and 8 repeats on his 2 alleles. His affected wife looks to be homozygous for 11 repeats, because that's the only spike that's coming up. In comparison, if you look at the 3 children, the unaffected child is clearly inherited an allele from each of the parents, but the 2 affected children appear to have only inherited an allele from the father, not from the mother. So one explanation might be a deletion, but we think we'd already excluded that possibility. But the more likely explanation that it took us an embarrassingly long time to think of was a repeat expansion that was so large that we simply couldn't amplify it using the standard technique. And so we simply weren't picking up the mutant allele using the standard methodology. And so to confirm that that was the case, we had to resort to repeat prime PCR combined with southern blot analysis. And using repeat prime PCR, samples from normal controls and unaffected family members, all of whom have fewer than 25 repeats in each of their alleles, they show this normal pattern on the electropherograph with a defined cutoff. It sort of ends rather abruptly. whereas all of the affected family members show what's referred to as stutter amplification, where you get this never-ending series of spikes. It just keeps going on and on. You blow it up and there's more and more spikes. All that tells you is that they have an expansion too large to be detected by this system. It's beyond the detection limit, which is about 50 repeats. So it's not quantitative. It doesn't tell you how many repeats. It just tells you there's too many, or that there's more than the control population. So then we had to try to remember how to do a southern blot, which was challenging. But what we found was that the samples from the affected individual, shown in lanes 3 to 6 here, in addition to the wild type allele at the bottom, they all showed variably sized expanded alleles, which we estimated in this small sample to range between 700 and 1600 repeats, so massively expanded alleles. Our study was published back-to-back in October 2011 in Neuron with another similar study, a group that we've been sort of communicating with. And in those two studies, and in numerous studies published subsequently from many centers around North America and Europe, it was confirmed that the C9orf72 mutation is the most common genetic cause of familial ALS, of familial FTD, a significant cause of sporadic forms of both of these diseases, and the explanation for the vast majority of families where FTD and ALS coexist. The phenotype is quite variable. The ALS tends to be classical ALS with early involvement of both upper and lower motor neurons, slightly earlier onset, slightly shorter duration than non-mutation cases, a higher prevalence of bulbar onset. About a third of the patients present with bulbar onset. But the biggest difference is the much higher frequency of dementia in the patients or other family members. In familial FTD, the C9orf mutation is more common than either granulin or MAPTau mutations. Usually presents with behavioral variant FTD, sometimes progressive non-fluent aphasia. But there are a variety of other presenting features that are relatively common. Quite a lot of memory disturbance, a lot of Parkinsonism. Up to 40% of our patients have some degree of Parkinsonism. And particularly in series reported from the UK, a very high presentation of major psychiatric illness, often very severe physical delusions, people describing big chunks of plastic stuck in their head and radio transmitters inserted in their anus, and all kinds of really interesting things that you don't usually hear in most FTD. An important aspect of this, and I think a general theme in the familial FTD, is that there's a lot of intrafamilial variability in the clinical phenotype. So as I was describing earlier, we've got families with two siblings, one of whom develops ALS at age 36, and their sibling develops FTD at age 65. You know, same gene, share 50% of the same genetic material, and yet that tremendous degree of genetic heterogeneity. In addition, I think this was an important discovery not only because of how common the mutation is, but also it introduced a new genetic mechanism to the fields of ALS and FTD, which again is why it took us so long to clue into this, is because I'm not a molecular geneticist, but even the geneticists who were working on this weren't very familiar with this type of genetic abnormality, which is these non-coding repeat expansion disorders. And there's a growing list of neurological disorders that have this sort of genetic mechanism. And they tend to occur by one of two different general mechanisms. In some conditions, there's an apparent loss of function mechanism. And this occurs when the repeat expansion interferes with the promoter region of the gene, silences transcription, so you get knockdown of the messenger RNA and decreased levels of the protein production. And usually these disorders are autosomal recessive because you have to knock out both alleles in order to sufficiently suppress the protein. However, there's a larger group of cases which are autosomal dominant, in which there's no problem with transcription, no problem with translation. You get normal amounts of the normal protein, but instead there seems to be a gain of toxic RNA function. And this occurs because when the pre-messenger RNA is converted to messenger RNA and you cut out the non-coding portions, the mutant RNA that contains that massive repeat expansion can't be degraded, and it accumulates in the cell as these so-called RNA foci that have the potential to bind and sequester crucial RNA binding proteins. And it's apparently the reduction of these RNA binding proteins that then have a negative effect on the splicing of other genes. And so it has a sort of downstream effect on other gene activity. And in our initial publication, we provided evidence that both of these potential pathogenic mechanisms were at play with the C9orf72 mutation. And this is because C9orf72 has three transcript variants shown here as variant 1, 2, and 3 different RNA transcript variants that encode two different length protein isoforms. The hexanucleotide repeat, shown as this red diamond, is located between two alternately spliced first exons. And so what that means is that in variant 1 RNA, it's actually located in the promoter region, where expansion silences transcription. We've shown that with this TACMAN assay. Silences transcription, so you get knockdown of variant 1 messenger RNA, with the predicted effect being reduced production of the long protein isoform. But in addition, in variants 2 and 3, the repeat is actually within the first intron. So here you have the possibility of generating RNA foci. And we confirmed using FISH with a complementary RNA probe that in fact there are these RNA foci shown as these little red dots within the nuclei of 25% of cells in the frontal cortex and the ventral gray matter of the spinal cord in our mutation carriers that are not there in non-mutation carriers. Now the effects of protein deficiency largely depend on the normal function of the protein, which we currently know nothing about. So C9orf72 stands for chromosome 9 open reading frame 72, describing its chromosomal localization, but we know nothing about its function. It has no known functional domains. It does show fairly striking structural homology to the DEN family of proteins, which are a group of proteins involved in various aspects of membrane trafficking and mobilization, so the formation of lysosomal and endosomal vesicles, autophagocytosis, these sorts of processes. And so it may be that C9orf72 serves a similar function, but at the present time we don't know. Commercial antibodies are available against the protein. They're not well characterized, so I'm not sure how valid these findings are, but this is some initial immunohistochemistry I did. What we tend to find is that the normal staining pattern is a fairly moderate diffuse cytoplasmic staining of neurons and this light punctate staining of the gray matter, which usually suggests a synaptic localization. And what we find in cases with ALS is increased staining in chromatolytic neurons and some swollen axons. But you find this in all cases of ALS, both with and without the mutation. And so we think this is just a non-specific feature of impaired axonal transport, basically. And in the hippocampus, in cases of FTD, pyramidal neurons show this very coarse punctate staining on their surface, suggesting expanded presynaptic terminals. But again, this is not mutation specific. You see this in all cases of FTD and even in a lesser extent in cases of Alzheimer's disease. So the important thing, though, is that we do not see evidence specifically for reduced protein levels, although this is a very blunt instrument for trying to demonstrate that, nor do we see any abnormal accumulation in the form of inclusion bodies or that sort of thing. The pathology associated with the C9orf72 mutation is fairly consistent among cases. Those that present with FTD show FTLD TDP with the typical TDP positive inclusions in the cerebral cortex and limbic regions. Cases that present with ALS have the classical pathology of ALS, degeneration of upper and lower motor neurons with TDP positive inclusions in the lower motor neurons. Importantly, the overlap in the pathology is even greater than the overlap in the clinical phenotype. And what I mean by that is patients who die with clinically pure FTD are almost always found to have at least mild TDP pathology in their spinal cords, and patients who die with clinically pure ALS always have TDP pathology in their extramotor neocortex and hippocampus. And what that implies is that the environmental and genetic factors that influence the phenotype are only relative, and that all of the patients who carry these mutations are probably susceptible to develop both FTD and ALS. In addition to the TDP pathology, an absolutely specific finding for these cases is that they also have additional pathology that can be demonstrated with ubiquitin immunohistochemistry that is negative for TDP, including these large sort of fuzzy cytoplasmic inclusions in pyramidal neurons of the hippocampus and these more compact inclusions in the granule cell layer of the cerebellum. And this ubiquitin positive TDP negative pathology is absolutely sensitive and specific feature for these cases, to the point that I can predict so far with 100% accuracy. And in fact, I've had a number of cases where the genetics have said, no, there isn't an expansion, and I've said, yes, there is, and it's turned out that, yes, there was because of a faulty genetic testing. So this is absolutely sensitive and specific. And what it implies is that there is mismetabolism of some additional protein that is not TDP and is not C9orf72. And 2 years ago, two groups, Dieter Edbauer's group in Munich, Len Petruccelli's lab at the Mayo Clinic, showed that this ubiquitin-positive TDP-negative pathology in the C9orf72 cases was the result of unconventional repeat-associated non-ATG-initiated, or RAN, translation of the hexanucleotide repeat. So even though the repeat is in a non-coding region of the gene, when it is massively expanded, it can trigger translation through aberrant genetic mechanisms that I'll describe in a minute. And so translation of the sense transcript in the 3 alternate reading frames is predicted to generate 3 different 2-amino acid dipeptides, glycine alanine, glycine proline, and glycine arginine. And in these initial studies, both of these groups generated novel antibodies against these different dipeptides and showed that they labeled that ubiquitin TDP-negative pathology absolutely sensitively in the mutation carriers and showed nothing in the non-carriers. And then subsequently, both of these same groups showed that this RAN translation also occurs in the antisense direction, which generates additional glycine proline as well as 2 more dipeptides, proline arginine and proline alanine. And so this introduces a potentially, or a third potential pathogenic mechanism to these cases, the potential toxicity of this dipeptide repeat pathology. RAN translation was initially discovered in cases of spinocerebellar ataxia type 8. It can occur when a repeat region forms a hairpin configuration that acts as a sort of surrogate start codon, and is more likely to occur with very large expansions, and particularly ones that are rich in C's and G's through CG cross pairing. And it's now been shown to occur not only in SCA8, but also in myotonic dystrophy type 1 and fragile X-terminal ataxia syndrome. However, in none of these conditions is there strong evidence that the products of RAN translation directly contribute to disease, that the products are toxic. And so to investigate the potential toxic role of the dipeptides in the C9orf72 cases, Manuela Neumann and I recently conducted a large clinical pathological study of 35 cases with the C9orf mutation, including cases with pure FTD, clinically pure ALS, and a combination of the two. We used an anti-polyglycine alanine antibody from Dieter Edbauer, which is very sensitive for picking up the pathology and absolutely specific for the C9orf72 cases. And what we found was that in the neocortex, this antibody labels neuronal cytoplasmic inclusions that appear as these aggregates of coarse granules within the cytoplasm. This is the most common type of inclusion. To a lesser extent, you get more diffuse granular staining of the cytoplasm that we refer to as pre-inclusions, because we think it's just an earlier stage before they condense. And in addition, I don't know whether this projects, but you also frequently get these small dot-like intranuclear inclusions. In the neocortex, this pathology has a somewhat laminar distribution, probably doesn't project well, but it's concentrated in layers 2 and layers 4. But importantly, it is equally abundant in all neocortical regions. There's just as much in the occipital cortex as there is in the premotor frontal cortex. In the hippocampus, you see abundant dipeptide inclusions in the granule cells of the dentate fascia and in the pyramidal neurons, so this corresponds with what I was showing you earlier with ubiquitin staining. In the cerebellum, in addition to the granule layer inclusions shown here, you also get lots of cytoplasmic inclusions, intranuclear inclusions, and these little neurites in the molecular layer, and occasional inclusions and pre-inclusions in Purkinje cells. The pathology in the basal ganglia and the upper brainstem is more variable and more modest, but here, for instance, is a pigmented neuron in the substantia nigra with a little cytoplasmic inclusion. However, you very rarely get inclusions in lower motor neurons in the medulla or the spinal cord, even in cases with clinical ALS. And so when you look at the severity and the anatomic distribution of this pathology across anatomic regions, there is it's absolutely consistent. There's no correlation with the various clinical phenotypes. And so you have abundant pathology in the neocortex, the hippocampus, and the cerebellum, moderate pathology in the basal ganglia, and minimal pathology in the brainstem and spinal cord. Using double label immunofluorescence, Manuel and I showed that the dipeptide inclusions shown in red and the TDP43 positive inclusions shown in green almost never co-localize to the same cell. They can be in the same general anatomical region, but they don't occur in the same cell, suggesting that there isn't a direct relationship that one causes the formation of the other. And when we look at the clinical pathological correlation in more detail, what we see, first of all, is that the pattern of neurodegeneration, so neuronal loss, reactive gliosis, microglial activation, closely matches the phenotype, as you'd expect. So in cases of ALS, there's severe degeneration of the spinal cord, there's severe degeneration of the primary motor cortex, whereas in cases of FTD, the most severe involvement is of the premotor frontal cortex, and then the ALS FTD cases have a combination of the two. The pattern of TDP pathology very closely mirrors that of neurodegeneration, but the pattern of dipeptide pathology is all over the place. It's consistent across the phenotypes, and it shows lots of pathology in areas like the occipital cortex and the cerebellum where you rarely get any degeneration, and it shows minimal pathology in areas such as the spinal cord where you may get severe degeneration. And when you just pool all of the data, the entire cohort, again, there's a strong correlation between the TDP43 burden of pathology and degeneration in these key anatomic areas, and no correlation with dipeptide pathology. So based on that, in our initial study, our conclusion was that neurodegeneration in these cases is more closely linked to the mismetabolism of TDP, and that the dipeptide pathology is a very sensitive marker for the present and specific marker for the presence of the mutation, but that its role in pathogenicity was really very uncertain. Probably not much evidence for it. Now, subsequent to that, a number of studies have provided in vitro evidence for a direct toxic effect of dipeptide expression, even in the absence of TDP pathology or RNA foci. Each of these studies use slightly different model systems. Two of them claimed that it was the arginine-rich dipeptides that were toxic. The other two claimed that it was glycine-alanine. And all of them implicated a different pathogenic pathway. So the relevance of this to human disease, I think, still needs to be sorted out, but it does raise a number of limitations to our previous human study, in that we only looked at glycine-alanine because at the time that was the only good antibody we had, and it may be one of the other dipeptides that's more toxic. We didn't look very closely at the specific kind of inclusion, and it may be that the nuclear inclusion is more important than the cytoplasmic inclusion. One of these studies, forgotten which one here, claims that it's the binding of the dipeptide to the nucleolus that triggers the toxic event. And of course, we only looked at the insoluble protein dipeptide species. We didn't look at the soluble species. So we're just in the process of completing a follow-up study in which we used antibodies against all 5 of the dipeptides, each of which labels at least some of this pathology in these cases in a sensitive and specific fashion. And a couple of interesting findings. First, the amount of pathology demonstrated with each of these antibodies is quite variable, and it's variable in a consistent fashion, meaning that the same antibodies show more pathology in different anatomical areas and amongst different cases. So what you can see here is the total number of inclusions stained for each of the antibodies in the frontal cortex and in the spinal cord for each of the cases. So this is all of the cases pooled. Each dot represents one patient in our cohort. First thing to point out is the difference in the y-axis. So for the frontal cortex, this is actually a logarithmic scale that goes from 0 to 1,000 inclusions per square millimeter, showing you how much more abundant the pathology is in the neocortex. Whereas in the spinal cord, this is a linear scale that only goes up to 10. So there's a vast difference in the burden of pathology. But interestingly, if you look at the hierarchy of sensitivity of the antibodies, it's the same in both cases. In general, the antibodies that recognize the products of the sense transcripts, so the glycine-containing ones, tend to show more pathology than those demonstrating the antisense products. Whether this is a true reflection of the differences in the amount of protein burden or whether it simply relates to the antibody sensitivity, we're not sure, but it's at least consistent across all cases. We conducted an enormous number of clinical pathological correlations. We looked at every type of inclusion, neuronal cytoplasmic, intranuclear, pre-inclusion dystrophic neurite, stained for each of the different 5 dipeptides in each of the different anatomical areas, correlating with different clinical features, presence or absence of ALS, presence or absence of FTD, age of onset, duration of disease. The only correlation that even came close to significance was earlier age of onset of disease was related to a heavier burden of glycine-alanine pathology in the frontal cortex. That was the only one, and even that just barely reached a fairly lenient cutoff for sensitivity. When you look, as I'm showing here, at just the overall pattern of protein accumulation in, again, the frontal cortex, spinal cord, cerebellum, in each of these different clinical subgroups, we saw no difference in the pattern, again, of the burden of pathology across the different clinical groups for any of the types of inclusions, for any of the different dipeptides. So in cases of ALS, shown in this top row, they have no more inclusions of any morphologic type or composed of any dipeptide in their lower motor neurons than the cases without ALS. So we're back to this point where we think that the dipeptides are a very consistent feature of these cases, but that we do not have evidence from human disease that they have a direct role in neurodegeneration. So to summarize what we currently know about the pathogenesis associated with the C9orf mutation, it's associated with 3 potential pathogenic pathways: reduced protein production, the generation of RNA foci, which is actually what I favor as being the crucial pathway, the RAN translation generating the peptide repeat pathology. The relevance of importance of these 3 pathways is currently unknown and clearly requires additional investigation. So to wrap up, I think in the last decade we've made tremendous advances in our understanding of the molecular basis of FTD. This is the current sort of classification as I see it, at least. I believe we've discovered all of the common FTD causing genes. There's still a lot to be learned about the genetic modifiers, and there may be some small genes left, but in fact all of our autosomal dominant families that we have at UBC are now solved, with one exception. And I think we've actually just found a new gene that explains that one family. So I don't think there are going to be any more big genes, big FTD genes discovered. All of the major pathological proteins have been discovered. However, we clearly still have a lot to learn about the pathogenic pathways that these genes and these proteins trigger, which is going to be crucial to developing useful therapies. Nonetheless, I think these discoveries have brought us much closer to advancing patient care and are already actually being translated into improved patient care in ways I'll describe in just a second. The one thing that's really, I think, holding back clinical advances in this field is the fact that although there are some fairly specific clinical molecular correlations, so for instance, patients that you see who have semantic dementia are almost always going to turn out to have TDP pathology. Patients who present with a classical PSP syndrome almost always turn out to have tau pathology. Most patients with ALS and FTD turn out to have TDP pathology. But there's a tremendous amount of phenotypic overlap, meaning that it is very difficult, simply based on the clinical features of a patient that you see, to be able to predict the underlying molecular defect, unless they have a gene that you know. But if that's not the case, then, to be honest, it's really a bit of a crapshoot here, which really highlights the needs for biomarkers, for biomarkers, in vivo biomarkers that allow us to predict the underlying molecular pathology. Because I think if we're going to make advances in treatment, they're going to have to be molecularly targeted treatments. We're not going to come up with a magic bullet that salvages frontal neurons or lower motor neurons in a fairly broad fashion. And so it was at least somewhat optimistic or reassuring that at the recent International Conference on FTD that we hosted in Vancouver just a few months ago, there were a number of presentations describing the results of new biomarker studies. So for instance, the tau imaging ligands that are now available and starting to be used, new biofluid markers for measuring TDP43 levels in various biofluids. And there are already a number of clinical trials that are started looking at targeted therapies for specific subtypes of FTD. So there are 3 different progranulin replacement trials currently running that I'm aware of. So although we've still got a long way to go, I think we're certainly taking fairly major steps in the right direction related to FTD. So I'm going to stop there. I'll thank my local collaborators at UBC, both our FTD research group and our ALS clinic, as well as our international collaborators, particularly Manuela Neumann in Tübingen, Germany, who I do a lot of my pathology work with, and Rosa Rademakers at Mayo Clinic Jacksonville, who does most of our molecular genetics. Thank you very much. Any questions?
I'll ask it. So you're not so enthusiastic about the pathogenicity of those dipeptides. Do you think, what do you feel about the use of them as a biomarker for them?
I don't think, I mean, based on my current understanding, I don't think they would be of any use as a biomarker. They would tell you that the patient has a mutation, but it's easier to prove that using standard genetic methodology. I mean, so as an example, I've got 2 cases that I'm aware of patients, family members with C9orf mutations, in which a family member died prematurely. One of them died at age 25 of a pulmonary embolus. From a dipeptide point of view, brain looks exactly like his mother, who died at age 60 with full-blown FTD. No difference in the burden, the distribution, the amount. This is something that starts early, somehow reaches a certain level, and stays there. And I find it hard to believe that that is the basis of neurodegeneration. And I also doubt, based on that little bit of evidence from a couple of cases, I doubt that it's going to fluctuate in any way in accordance with the disease so that you could use it even as a marker of, you know, a drug having a positive effect or not. Having said that, the people who are proponents, the Len Petruccellis and the Ed Deepbauers, keep producing in vivo and in vitro models suggesting that there may be a toxic effect, but I'm skeptical. I mean, you can kill neurons with anything if you just hit them hard enough. Yep.
Related to that, these dipeptides form through this REN mechanism,
Yep.
is that right?
Yep.
And you said that that was more common the longer the repeat?
Yes. Just in general terms. So if you look at different diseases with different kinds of repeats, so the SCAs and the other ones, the ones that tend to have this RAN translation going on are the ones where you get very large repeats. Not the 30 and 40 repeats, but the thousands repeats.
I was surprised then to see other diseases on your list of Fragile X tremor ataxia, but not Fragile X then.
But it's a different mechanism in the 2 processes, which perhaps Tom can explain better than I can. But in the Fragile X tremor ataxia syndrome, where you've got these sort of pre-mutations, their mechanism of disease seems to be through this toxic RNA gain of function, whereas the Fragile X syndrome is a distinct mechanism. But I guess what you're asking me then is, it should be the other. You would expect that if the RAN translation came into play, it would be more likely to come into play with the Fragile X patients because they're the ones with the longer. But I don't think either of them. Well, yeah, I'm not sure how to answer that. You're right. Yeah. Right. Right. Okay.
One more question. I was looking at your table. I recently had a patient with dementia, ALS, and Parkinsonism.
Mm-hmm.
And I was just trying to think, what genes would you think of as more likely to be affected in that situation? All 3.
Sorry, I lost my sight here. So it could be, of the genes, the most likely one would be C9orf72, largely because it's the most common of the genes. It's the most common one that would cause dementia. Sorry, did you say dementia, ALS, and Parkinsonism? About 40% of them develop some degree of Parkinsonism. And so that would be the most obvious one. The other possibility, if it was a sporadic case, would be one of the FUS-based ones, so either NIFID or BIVD, very uncommon disorders, but their phenotype typically is early onset FTD combined with Parkinsonism, and usually the motor neuron disease is actually a PLS more often than an ALS. But that would also be a possibility. Tau can potentially do that. Tau can certainly cause the FTD and the Parkinsonism. The typical ALS, I think, is uncommon, although you can get a sort of amyotrophy, I think, with some of the tau mutations. Is that right, Tom?
Could I put in a marketing advertisement?
Sure.
Are you aware that University of Washington genetics commercial lab now has a panel for exactly that kind of patient. It includes all the genes you've been talking about.
I was not aware of that.
There you go. That's my advertisement for that. It's available. It's there. You just order it. And
The one counter advertisement for that is we have something similar at UBC. At least Matt Farrer has a chip with, I don't know whether it's 200 or 400 different neurodegenerative genes on it now. I feed him cases. Every one of them comes back with at least 20 or 30 polymorphisms, and you scratch your head and wonder, what on earth has presenilin got to do with this? You know, what on earth does dynactin have to do with this? And so you get a lot of information. There's a lot of variability there, and being able to sort it out. If you can find what you're hoping to look for and it makes sense, I think it's great. So if you happen to find exactly as Tom says, if you're trying to decide it could be this, this, or this, and it tells you it's this, then great. But if you just got a weird case and you're looking for a gene, it's really hard to sift through that data unless you can take that and then show segregation within the family and sort of prove its pathogenicity. Right now we're just having trouble with the informatic aspect of that kind of thing.
there's also a little asterisk to that, unfortunately, and that is you got to be sure and ask them to do C9 or 72 separate.
Right. Because that's not something that's going to be picked up on a natural screen. You're not going to get that.
I asked them to do that one, and they refused to do any testing. They didn't want it. She's died now, so.
So TDP43.
Yep.
Am I right that it's very non-specific? I mean, you can see it in Alzheimer's, human Alzheimer's.
Yep.
Is that right?
Sure. So it's I guess that means it's non-specific, in the sense that tau is non-specific or that sort of thing. So you do see it you see it as the primary or the sort of exclusive signature pathology in certain diseases, largely ALS and subtypes of FTD. But you're right, you also see it as a common co-pathology in many neurodegenerative disorders, but usually with a very restricted anatomic distribution of the mesial temporal lobes. So if you look, as we've done at a large series of Alzheimer cases, for instance, and many people have done this, somewhere between 25 and 50% of them will have some TDP pathology. Starts out in the amygdala, then you see it in the entorhinal transentorhinal cortex, then you see it in the pyramidal layer, and then it usually stops. Very rarely do you ever see it into the neocortex in a pattern that would be typical of FTLD TDP. So in that situation, I believe that that's a secondary phenomena that's occurring as a result of the primary Alzheimer's disease. You can also see it in Pick's disease, you can also see it in Lewy body disease, but it almost always has that same distribution. And so I think those populations of mesial temporal lobe neurons have used TDP or metabolized TDP in such a way that when they are stressed by their primary disease, you also can get TDP positive inclusions in those cases. Yeah. So you're right, it's non-specific in that sense. I took the lack of sensitivity and specificity only in the direction of the non-TDP cases, but the other way you could take it is TDP expanding into some of these other disorders. You're right. Yeah. And one thing that is important in trying to figure out how this fits together is that as part of a normal stress response, TDP translocates from the nucleus into the cytoplasm and starts doing a number of things. It helps move proteins into stress granules, RNAs into stress granules to sort of protect them temporarily. It also transports RNAs to places where they're needed for sort of repair. So it transports actin to dendritic terminals, for instance, to help repair that part of the cytoskeleton. And so one possibility is that a fairly common theme here is that in the diseases characterized by TDP pathology is that there's some triggering insult. The cells try to respond by a TDP43 response, shove it out into the cytoplasm to get it to protect the cell or repair the cell. But if that insult isn't temporary and it persists and persists and persists, and TDP hangs around long enough in the cytoplasm, it begins to misfold, mismetabolize, aggregate, fibrilize, and then take on some adverse toxic effect. It starts binding RNA proteins that it shouldn't be binding. It starts sequestering things. So there may be a common theme there. The fact that mutations in the TAR-DDP gene directly cause ALS, at least is some evidence that it has a primary role in disease. but it's harder, for instance, in these cases Tom mentioned, the Alzheimer cases, where it seems to be more of a secondary, the pathology is more of a secondary effect.
Thank you very much.
Anything else? Thank you.